Okay, so adaptive immunity or specific immunity uh, protects us against things that are gone wrong either in our own body cells or by some pathogen, some infectious agent. Um, it works with the non-specific defenses. Uh, some things it works just in conjunction with, and other things it's more specifically works with. For instance, inflammation is enhanced by what happens in the specific responses, and complement needs that specificity to activate it, and so the two work hand in glove in that case. Fever and other things are involved as well, but they're the non-specific. So we're going to talk about the specific things. Um, so I, although I said this in class, I figured I'd go back and start at this point again. So the thing about the adaptive immune response is that it's very specific to a specific pathogen or a specific problem. It's systemic, so you have a body-wide response, and it has memory. And there's two parts to it. There's humoral immunity, which is mediated through antibodies, and then there's the cellular, cell-mediated immunity. In the humoral immunity, the antibodies are what does the action. It is what challenges the antigen. Um, these are non-living extracellular proteins that are made by cells of the adaptive, but the agent is this extracellular thing called an antibody. In the cellular immunity, the cells themselves interact with the antigen and are the agents of change. Now, it's interesting, in specific immunity, really the whole point of specific immunity is to eliminate the antigen. So however it goes about it, it's about eliminating the antigen. The antigen is that which generates the immunity, the immune response. So it generates anti. So it's anything that can evoke this immune response and mobilize the defenses. They tend to be complex molecules, they tend to be large molecules, and oftentimes they're organic. They are anything that is non-self. To be an antigen, you have to have immunogenicity, so be able to stimulate the adaptive response, the lymphocytes, and reactivity. They have to be able to react with the antibodies that are released. So all kinds of things fit this category. Foreign proteins and polysaccharides are the two most important ones. This is non-self. It's interesting, I'll just throw this in here. If you get a sliver of metal or glass, you get some inflammation, you get a lot of things going on. But if you get a sliver of wood, you also get a specific response against that, and that's when you get the pus and much more of it, because wood comes from a living organism, and so has these antigens in it where glass and metal don't. There are some things called haptens, which are half antigens that do not have immunogenicity, but they have reactivity. These things bind to proteins that are made in the body to become complete and to get immunogenicity. So if you don't make the proteins for it, then you probably won't have a reaction for it. Poison ivy, animal dander, that kind of thing are the classic examples of this. I don't make the protein that poison ivy needs to attach to in order to become antigenic, so I don't react to poison ivy, knock on wood. I also don't react to animal dander. My brother, on the other hand, uh, can't be in the same building as a cat and without having a pretty severe reaction. So he makes those proteins, I don't. So antigenic determinants, again, I spoke about this in class. These are the things that make the antigen react. These are the aspects of the antigen that are critical to this immune response. Some antigens have many different antigenic determinants, and some of them only have one or two. If there are a bunch of different antigenic determinants, which are sometimes called epitopes, then the reaction will be stronger than if there is just one. The example I used in class was your attraction to members of the opposite sex. What are the determinants that you particularly like? And the more of those determinants there are, the stronger the attraction. So most antigens have all kinds of these antigenic determinants, and will evoke a fairly large response, because these big complex molecules have all kinds of different places that can be antigenic determinants. Like a protein can have certain amino acid sequences and things. Large simple things like plastics, glass, and that kind of stuff really doesn't have very many antigenic determinants, if any, so is not immunogenicity. Again, we looked at this in class. This is an antigen with 3 different determinants that will react with 3 different antibodies, and so therefore it evoke a very strong reaction. Now, it's interesting because one of the things that has to happen is our immune system has to recognize what belongs to us, what is part of our body, and what is a foreign invader. We don't want to attack our own stuff. Sometimes that goes wrong, and those are called autoimmune diseases, things like rheumatoid arthritis and lupus, etc. But we need a way of identifying who's kind of on our team. Every cell in our body wears kind of a uniform. It has molecules on the surface of the cell that are antigenic to everybody else's immune system, but not our own. Our own immune system knows to ignore them. So we call these MHCs, which stands for Major Histocompatibility Complex. And everyone is a unique MHC. Now, people that you're closely related to probably have similar, but they won't be identical. Now, there's 2 classes of MHCs. Class 1 is the common, widespread one found virtually everywhere. They're on almost all body cells, and they basically are little flags that say, I belong here, please don't attack me. There's another type of class of MHC, they're called class 2. And these ones are a little bit more active. And we find them on cells that are involved in the immune response. Both of them contain proteins or protein segments that act as self-antigens, right? And the proteins that are incorporated in these MHCs, and the MHCs are constantly being made and reabsorbed and made and reabsorbed. So these things are very dynamic. The cell doesn't make its MHCs and then leave them. It's constantly making new ones. And it incorporates proteins from inside the cell. So if the cell is infected, then some of the infected proteins end up in the MHC and really identify the cell as being infected, say by a virus. Cells that are cancerous or cells that have something wrong with them will incorporate some pretty strange proteins into their MHCs and again identify that there's something wrong with that cell. We'll talk more about the class 2 MHCs in a couple of minutes when it's appropriate. Now, the adaptive system really relies on two different types of cells, and they're both called lymphocytes, or two general classes. There are B lymphocytes that are involved with humoral immunity. They're the ones that are involved with making antibodies. The B lymphocytes themselves don't attack the antigen. They provide the antibodies that attack the antigen. Excuse me. There are T lymphocytes, and they're called T lymphocytes because they're made immunocompetent in the thymus. They learn what they're supposed to attack in the thymus, and these are a lot like natural killer cells. They kill. They do the killing themselves. They kill specific infected cells or cancer cells. Now, in order for this to work, we need something called antigen-presenting cells. And what antigen-presenting cells really are is a warning system. It's what actually presents the antigen to the lymphocyte to evoke the immune response. You don't really want the pathogen to spread through your entire body and until it reaches the lymphocyte, the appropriate lymphocyte, and evokes the immune response. You would much rather have a macrophage eat the pathogen, incorporate parts of that pathogen, the antigenic parts of that pathogen, into its MHCs, and then have this macrophage roam around until it meets the appropriate lymphocyte and then activate the lymphocyte. It's much safer for you to do that. You want really the antigen presenting cells present evidence that there is a pathogen by just presenting the antigenic portions of the pathogen. This is really very, very, very important to getting this whole adaptive immune system started and up and running. You need these antigen presenting cells. Now, the lymphocytes all start in the bone marrow. They're the second most common, after neutrophils, of the white blood cells. Never let monkeys eat bananas. The lymphocytes that will become B lymphocytes mature in the bone marrow, and they learn, they get their receptors to their specific target while they're still in the bone marrow. The T lymphocytes, however, migrate to the thymus, and this is where they become immunocompetent. This is where they mature and become useful. Now, there's two things to maturing a lymphocyte. One is being able to recognize the antigen that they are specifically designed to meet and eliminate, because the whole point is to eliminate the antigens. But they also have to learn to ignore the self-antigens, the MHCs, because you don't want these things attacking your body willy-nilly. Um, so, so that's what happens, right? So these things are made in the bone marrow. If they're B lymphocyte, they become competent in the bone marrow. If it's a T lymphocyte, it becomes competent in the thymus. And then they're transported to the lymph nodes, to the spleen, to the tonsils, to other lymphoid organs that we've talked about in the past, where surveillance actually takes place, where these things can work. Now, when they're transported there, they're considered naive. They haven't met the antigen yet, so they haven't evoked a response. They're sent there for surveillance, to watch for their specific antigen. And that's where they'll go to work, and that's when they'll do what they need to do. So this is just a slide showing that. Made in the bone marrow, becomes mature, then are sent off to lymph nodes and other lymphoid tissues, the Peyer's patches and appendix and all kinds of places, any of that lymphoid tissue. So I'm not going to get too deep into what the T cells do at this point, but basically they are selected, right? And what they do is they have to kind of pass some tests. So they have to the thymus eliminates the T cells that are overactive to self-MHCs and kills them, right? So it looks for the ones that can possibly do us damage, and we won't bother with it beyond that. B cells, the bone marrow looks for ones that react to other self-antigens, and they're eliminated, right? They are inactivated somehow. And that's the point here is that as part of the maturation of all the lymphocytes is to make sure that they are not going to attack self-antigens. That's the important consideration. Now, the antigen receptors, there's all kinds of them, right? And we have a different receptor for every possible antigen. And it's done through coding by about 25,000 genes. This is how important it is. It's about 25,000 genes. But we end up with 1 billion different types. But it's basically, to simplify this idea, is that how many words can you make out of 26 letters of the alphabet? And how many sentences can you make out of those words? It's kind of the same thing. We don't need to have a different genetic coding for every single antigen receptor. We have classes of them, and we can make unique receptors that way. So, back to the antigen presenting cells, and this is kind of a good starting point to the response. The pathogen, or whatever is the antigen, has the antigen on it, ends up in the body. From there, the antigen gets engulfed by some sort of a phagocyte. Usually it is a macrophage. Sometimes it's a dendritic cell. It may be even a neutrophil, I think, but basically we look at them as macrophages. or they can be B lymphocytes becoming activated by bumping into the pathogen in the interstitial fluid. So an activated B lymphocyte can also act to present the antigen. Just a photograph of a dendritic cell. Okay. What their job is to do is to engulf the pathogen, present the antigen in their MHCs to the T lymphocyte. Now this is important because the T lymphocyte's job is to kill body cells that are either A, defective, or B, have an infection in them. They are infected by a virus, for instance. So it recognizes the cells to attack by their MHCs. The macrophage engulfing the pathogen will present on the MHC and and activate the T lymphocytes. So there's a dualism there, as opposed to the B lymphocyte, which will be attacking the pathogen in the interstitial fluid. It doesn't have to be a processed antigen. But it works both ways. So once the T cell gets activated by the macrophage that's eaten the antigen, then it releases chemicals that make macrophages more active. So it becomes a bit of a positive feedback on that. They support each other. So really, the adaptive immunity as a whole has the express purpose of eliminating the antigens. The antigens are those aspects of the pathogen that will evoke an immune response and react to that immune response. It is a systemic response, body-wide, that ends up having memory. It uses the lymphocytes, B lymphocytes for humoral immunity, T lymphocytes for cell-mediated immunity, and antigen-presenting cells, and it specifically removes non-self substances, antigens.